In this video, we are going to talk about how to load and visualize your CHESPA result. The result should already be loaded after you finish your analysis. You can also load your result folder using the load option. It should be saved in a folder called CHESPA in your Sparky folder. The results are listed in a window. You can rearrange the results by sorting the residues by residue number, the chemical shear differences of both reference or perturbation vector, or the absolute value of the fractional inhibition, or cosine theta of your vector angle. In our previous video, we used the perturbation vector 2 to 3 and loaded an inhibitor. As we can see here, the majority of the fractional inhibitions are negative as expected. You can highlight the residues by double-clicking any entry. The peaks corresponding to this entry will be centered in the spectra involved in the analysis. You can plot the chemical shift differences, the fractional inhibition, or cosine theta against residue number by simply clicking the plot button. All the x-axis are residue numbers, and the y-axis are either the chemical shear difference of your perturbation vector, chemical shear differences of your reference vector, the fractional inhibition, or cosine theta. You can save the plots in SVG formats by clicking Save to buttons, and can save it in anywhere you like. You will see that many bars are marked red. This is because that in many cases, some values are much greater than 1 or lower than -1, and it will effectively suppresses other bars. When such residues are significant, the perturbation analysis. So we mark them red and keep the y-axis ranges from -1 to 1 or 0 to 1. Lastly, you can visualize your results in PyMo by simply hitting the PyMo button and load the PDB file. You do not have to renumber your PDB file, as CHESPA-SPARKY will automatically recognize the sequence from your assignment and create a PDB file in your result folder. PyMo should automatically start if it is in your environmental path. If it doesn't, a command here is already in your clipboard. You can start PyMo manually. And paste the command. You can select all the residues of your interest. You can even select multiple residues by holding the Control key. Here we are just going to select all the residues with a significant inhibition. Once selected, you should type @p in your PyMo command line. And here we can see all the residues along with your CHESPA result of your fractional inhibition and cosine theta plotted in your structure. This concludes our demonstration of CHESPA. Thanks for watching.